Hello, my name is Elizabeth Taglauer, and I am a neonatologist in the Department of Pediatrics here at Boston Medical Center, and I'm happy to share with you today our study entitled Characterizing the Human Preeclamptic Pulmonary Developmental Niche through Placental Spatial Transcriptomic and Proteomic Analysis. The background for this study is focused on the disease of bronchopulmonary dysplasia, which is a disease of premature infants that continues to impact the health of our neonates worldwide. Preeclampsia is a placental-driven pregnancy disorder that causes maternal hypertension and a systemic vascular disorder. And several clinical studies have identified that preterm infants born to mothers with preeclampsia have a significantly greater risk of developing this lung disease of bronchopulmonary dysplasia, or what we will call BPD. So the hypothesis of my work is that preeclampsia causes a unique type of developmental lung injury that predisposes our infants to an increased risk of BPD. Because preeclampsia is a uniquely human disease, and in order to study the most translatable type of science we can to help our patients, research on human placental tissues is needed to more clearly define the role of preeclampsia in developmental injury. For the rest of the presentation, I will also refer to preeclampsia as PE. So the objective of our work is using different types of molecular and proteomic analysis, namely spatial transcriptomic and proteomic analyses. We're examining primary human preeclamptic tissues to identify secreted factors that may be involved in injury pathways in the lung. Shown here is a study overview of how we're approaching this. First of all, we are seeking to understand the different functional areas of the human placenta. First of all, by identifying external cellular components that could be contributing to the pulmonary developmental niche. And to do that, we're using spatial transcriptomics of placental tissue. And we're also looking at internal soluble components using proteomics of placental secretome and cord blood. And I'll show you more of that, how we're approaching that in additional slides. So figure 1 shows our spatial transcriptomic analysis. The general principle of spatial transcriptomics is that it analyzes the different gene expression between tissues at the level of cellular niches. For this technology that I used, the cellular niches were in the 5 to 10 cell resolution. So each dot on these graphs is showing a small cellular niche of that and comparing the gene expression of those cells in comparison between preeclamptic tissue and a tissue from a patient with gestational hypertension. I chose these two different samples because preeclampsia is my disease of interest. Gestational hypertension is a much more mild form of this disease that is not associated with the development of neonatal lung disease. So in comparing two samples, one preeclamptic and one from a gestational hypertension pregnancy, I identified 3 cellular niches that had interesting gene expression changes in preeclampsia versus gestational hypertension. These were fetal blood vessels, 2 different types of fetal blood vessel niches, and also an area of immune regulation at the edge of the placenta involving extravillous trophoblasts. Shown here in part C, also the beauty of spatial transcriptomics is that in those cellular niches that you identify for the gene expression of interest, you can also then map those back to a piece of tissue. So to orient you, there's maternal and fetal sides of the preeclamptic or any placental tissue, and the cellular niches of interest were indeed mapping back to appropriate and functional parts of the placental tissue, as shown here in red. We then analyzed what genes were different between preeclampsia and gestational hypertension. We used 2 approaches. One was a unsupervised approach using biologic pathway analysis, and we identified then in our niches we saw an upregulation of pro-inflammatory signals in our fetal blood vessel niches, and also a downregulation of a key factor, insulin-like growth factor, signaling in our extravillous trophoblast niche. So we're seeing some key areas that could be impacting the developing fetal lung that are being signaled by the placenta. We also then I conducted a supervised analysis looking at gene transcripts that were specifically upregulated in preeclampsia and those that had been previously identified as important or related to fibrotic lung disease or BPD. In our fetal blood vessel niches, we identified 2 immune driving factors, one interleukin 32, as well as CXCL1. These are involved in pro-inflammatory cytokine production and neutrophil recruitment. Both have been previously implicated in BPD pathogenesis. And in our extravillous trophoblast niche, we found a new and interesting target regarding iron regulation. And there's been recent literature showing that iron overload in tissues may be related to the pathogenesis of fibrotic lung disease. So this could be an interesting area for future investigation. So with identifying some interesting potential targets from our spatial transcriptomic analysis and using this concept to say that yes, we can identify pathways and genes in preeclamptic tissues that could be connected to lung injury, we then decided to expand our work into creating a repository here at BMC. BMC is a particularly valuable area to study for preeclamptic because we have a very unique population of high-risk pregnancies. This pie chart is showing the racial and ethnic demographics of our patients at BMC. We are also a very unique population as a safety net hospital. As you all know, many of our patients are recent immigrants experiencing housing instability, and within, again, racial demographics and socioeconomic statuses that have not been previously studied, particularly in the role of preeclampsia and lung disease. So my repository is unique in that I am targeting tissues from pregnant patients either with preeclampsia or preterm labor at a very specific gestational age or a stage of pregnancy from 26 to 32 weeks. These are the most high-risk patients that are most likely to develop BPD postnatally. I'm first collecting placental explants from the tissue and culturing them in transwell plates. This creates a conditioned media with secreted proteins from the placenta that we can then harvest and use for future analysis and also future in vitro studies. At the same time, I'm also collecting cord blood from the samples and using that to compare an analysis of what's circulating in the fetus versus what's being secreted by the placenta. Our initial studies are conducting proteomic analysis of these tissues. The next piece of data I'll show is pilot proteomic analysis of the first set of samples from our repository. And the clinical demographics are listed here. And we did have a lower birth weight in our preeclamptic versus preterm labor patients, signifying growth restriction and a pretty severe form of preeclampsia. Our pilot proteomics did identify, in our pilot proteomics, we were able to identify established proteins that are involved in preeclamptic pathogenesis. This was an important anchor for us to show that our data was indeed useful and then could be further expanded into additional analysis. These 2 soluble proteins are soluble FLT1 and soluble endocrine. These are, FLT1 is a decoy receptor for VEGF, and endoglin is a decoy receptor for TGF-beta signaling. And both of these proteins have been important in human preeclamptic pathogenesis, but have not yet been very well characterized in human lung injury models. So the figure 2 shown here is our proteomic analysis of these 2 key preeclamptic proteins, first showing S-FLT. We did see a higher signal of S-FLT in our preeclamptic versus preterm labor placental conditioned media. We also identified this in the cord blood, so higher in preeclamptic versus preterm labor. However, we did see an even stronger and more significant signal in soluble endoglin, which was interesting and novel to this type of analysis, that a predominance of soluble endoglin over S-FLT secretion signal in our samples could be indicating that we indeed have a unique look at a severe or a novel subtype of preeclamptic patients. So in conclusion, our placental spatial transcriptomics showed that when comparing tissues from a pregnant patient with gestational hypertension, there are specific cellular niches and preeclamptic placental tissues that have differentially expressed genes that are associated with pro-inflammatory signaling and a downregulation of insulin-like growth factor signaling. And these can be tied back to BPD and pathways identified for fibrotic lung disease. And in our pilot data from our placental proteomics, we identify well-established placental signaling molecules that are known to be involved in preeclampsia, and they were detectable in our placental secretome and our cord blood, demonstrating feasibility for future analysis. We're also highlighting that we have a unique high-risk population at BMC and that our repository will be valuable for future study. So in future directions, we are continuing to expand our prime repository with ongoing recruitment. We're targeting 20 to 30 patients per year. And this final data here, these graphs are showing that we don't have a difference in gestational age, so we're recruiting really in a targeted range. But interestingly, the birth weight percentiles of our preeclamptic patients is much lower than preterm labor. So we're already seeing a very valuable cohort here in that our infants are already growth restricted and again, identifying they have a severe form of preeclampsia. We are then not only analyzing these conditioned media and cord blood samples, but we are also taking our conditioned media and coupling it with in vitro laboratory analysis in partnership with the Cotton Lab at the Center for Regenerative Medicine, looking at alveolar epithelial cells as well as pulmonary endothelial cells derived from human-induced pluripotent stem cells, again keeping with the theme of human-based tissue and in vitro analysis. We're also partnering with Steve Abman's group at the University of Colorado to inject this conditioned media into the intraamniotic cavity of pregnant rats and looking at the impact of overall fetal lung development. In conclusion, I would like to thank the Alpert Foundation for their funding to initially create this repository, the BU CTSI for continued funding to expand our repository, and also Boston Medical Center Pilot Award that will also help me continue expanding the repository and conducting some of these initial analyses. Thank you very much.